introduce session 2 then, which is the bioinformatics session. So I'm very pleased that Steve Jones has agreed to moderate this session. You all know Stephen from the BC Cancer Agency and the Genome Sciences Center. And we have 2 speakers today, Wyeth Wasserman from CMMT, and who will be speaking on the CanEuCre project that he's been working on with Beth Simpson and colleagues, and the genomic resources of advancing therapies for brain disorders. And then we also have Fiona Brinkman, and Fiona's going to be speaking about the applied metagenomic work that they're doing for the watershed microbiome. So we're very pleased to have both the speakers. What we're going to do is we're going to hold the questions for each speaker, and we're going to have the panel come up, and Steve and the panel and Fiona and Wyeth will sit on the front, and then it'll be open to questions from the audience as well as Steve. I know has some questions for the speakers. So with that, let me go ahead and welcome Wyeth, and please help me in just welcoming Wyeth Wasserman.
So I'd like to thank Brad and the organizers for the chance to come here and talk to you a little bit about promoters and transcriptional regulation. It's a bit of a change from where we were this morning, so hopefully you've braced yourself with a good lunch break and you're ready to get going. But that said, right after lunch I know that you're going to be having some blood sugar issues and you may be falling asleep as we go along, so I just thought I'd start by telling you the end. And say what we're primarily interested in this project is developing tools that allow us to direct gene expression in a selective manner to different subregions of the brain. And what we're showing here on this slide are 3 of our promoters that we've developed over the course of this project, each of which has a nice, precise pattern of gene expression that is directing in the brain. And our hope is, as we go along, is that these tools can facilitate both basic research and lead to the development of gene therapy-based applications that would allow us to direct gene expression to the appropriate locations for the disease or the gene at hand. So we'll come back now through and talk about how one gets to this endpoint where you have promoters with selective patterns of expression and where we're going with the CanEuCre project. So I'll start by telling you what a promoter is. For those of you who are not molecular biology oriented, we'll do a quick orientation. I'll introduce you to the CanEuCre project and our partners within that. I'll talk to you a bit about maxi promoters and mini promoters and what those mean. And then, because of this project, we are having the opportunity to begin to explore some gene therapy. I'll talk to you a bit about what we're doing with AAV viruses and how we're working with mice being able to deliver gene expression in the body in an efficient manner. So, this is the slide that's closest to my heart. As a computational biologist, I'm particularly enamored with the issue of how genes are turned on and off and understanding how cells achieve a different pattern of gene expression out of one genome. And when we go in and we look at the genome and we look at the gene may be turned on in a selective manner. The blue gene might only be expressed in the hair follicles, for instance, and the red gene might only be expressed somewhere in the eye. And we'd like to understand how that's possible and what are the key molecular sequences that direct that expression. Now, within this project, we're going to assume that most of that's occurring at the transcriptional initiation level. There's a lot of other levels that can be participating, but our focus will be on transcriptional initiation. And we're going to be in particular looking at how transcription factors interact with the DNA to direct that expression. Now, I'm going to hide under the hood an awful lot of things that are going on here. I'm going to hide under the hood the epigenetic properties. I'm going to hide under the hood chromatin effects. I'm going to hide the positional effects of where these sequences are located. And we're going to sort of take the candidate sequences that we work with and we're going to scrunch them down into a small piece of DNA that we're going to test. And that's not always going to reproduce what we want. Sometimes it might give us something better than we were going for. Sometimes it might give us some off-target expression. But from a perspective of a brain tool development project, anything that directs expression in a selective manner in the brain becomes a useful product of the research. Now, for promoters, we use the term promoters in a very encompassing way for this particular project. For those of you who are sticklers for this, a promoter might be just that small piece of DNA right at the beginning of the gene. For us, we're going to take all of the necessary sequences for directing the transcriptional initiation of the gene in a selective manner. So there might be enhancers in here, there might be regulatory sequences that are far away, there might be introns, 3' of the gene, 5' of the gene. So they could be anywhere. And we're going to do our best to focus on those cases where we have a better chance of finding where they're located. Now, this is a brain-oriented project. At the CNMT, we are particularly interested in a variety of disorders of the brain. We have interest in, for instance, Huntington's disease research, the development of brain stem cells and their properties. And so we've come to believe that we're going to need new mechanisms for getting into the brain and delivering therapy, particularly for diseases that are a lifelong disorder. And it's going to be hard over time to keep delivering drugs past the blood-brain barrier for an extended period, and we will probably need new genetic therapies in the long term to be successful. So, in order to do that, we would like to come to the time that we can direct that expression in the brain to the right types of cells, so that we're not turning on these therapeutic genes everywhere in the brain, but that we have sort of regional enrichment to the right locations. So if we're successful with this, we hope that we can therapeutically deliver gene expression to the target cells of interest, that we can minimize the off-targets gene activity, that these tools should facilitate basic research in a fundamental way, and that we could enable long-term the development of genetic therapies for brain disorders. Now, gene therapies have a bit of a history over time, but right now we're re-entering a new golden age for gene therapy. There's a lot of trials going on around the world showing that gene therapy can be successful, particularly right now in eye disorders, but others as well. But when we go in and we look at what's happening in those systems, the gene therapy promoters that are being used are by and large ubiquitous in their expression pattern. So they'll just turn on in whatever cell those pieces of DNA arrive to. Oftentimes those sequences are viral in nature, and cells are very good at shutting off viruses, and so they tend to get shut down over time, which makes long-term therapy a challenge. And they are relatively few of them that have used successful gene therapy up till now. And so we become interested in finding better ways to make promoters for delivering to these regions. We'd like our promoters to be selective in expression, as I said. We would like them to be human sequence. If we're going to do therapies to humans, we'd like them to be human sequences under the hopes that they will be more resistant to the mechanisms of the genome for shutting them off. We would like them to come from our endogenous genes, which means we're going to have to understand and look at the genome in a careful way. And we're going to be doing it using a larger scale pipeline than has previously been performed. Now, we're going to start at the midway point. So this is a project that's been going for a little bit of time now, and we had previously the Pleiades Promoter Project that was funded by Genome Canada and Genome BC. And that successfully ran to conclusion. There's a nice paper in PNAS which describes that effort and introduces the first 27 of the many promoters that we've created for the work. And it describes some of the protocols that we use in some of the bioinformatics as well. Now, coming out of that project and wanting to keep going and moving a little bit more towards tool development, we were looking for partners that we could bring on board that would make our promoters be more widely useful. And we developed a very good relationship with the International Knockout Mouse Consortium. So this is a large-scale effort. It includes legs in the United States and in the European Union and in Canada with Jeff Higgs, Stanford Summit, and others. And what they're trying to do in their project is to create a systematic introduction of knockout sequences in the genome of the mouse, such that if they introduce a gene called CRE, they can take out a gene in a selective manner. And so up till now, they're coming up to the point where they have about 20,000 genes with these sequences in place that would allow those genes to be knocked out. But in order to knock them out, they need this CRE enzyme to be turned on in the organism. And so CRE's been around for a long time. Basically, it recognizes an extended DNA sequence called the lock site. When CRE activity is present in a cell, it removes the sequences that appear between two lock sites, circularizes that piece and takes it away, leaving within the genome just the lock site. And so if you flank a piece of a gene with the lock sites, and then you introduce CRE, you can knock out the gene. And so the IKMC has been working on this for a long time, and they've got a systematic design. Systematic design here, and you can see that they've basically flanking an exon that's critical for the gene with lock sites, And then if you introduce CRE, it removes that key exon and you're left with a debilitated gene. But the problem for that group is that they just don't have very many sequences, many ways to drive CRE expression in an interesting manner. So they've got all these 20,000 mice that they can work with that have these sequences next to the exon, but they have a very small number of promoters. And so that led to our recognition that they had a bunch of CRE lines, a bunch of mice lines ready, waiting for CRE, and we had a bunch of promoters that we've been working on to direct expression. And so it was natural for us to come together under the CanEuCre project, which is a Canada-European Union group working on the development of CRE resources. And this has been funded by Genome BC, along with the European Union funding the Contours project. Within our team, we break it down into a few activities. I'm the computer geek, and I sit there and try to design the DNA sequences that we test. Rob Holt from the Genome Sciences Center works on large-scale DNA manipulations and the construction of our maxi promoters, which I'll talk about in a moment. We're led very well by Dr. Elizabeth Simpson, who's in the back today, who leads the mouse and the cell-based work. And then when we have a brain, or in this case other organs as well, Dan Goldowitz is the anatomist who looks through histology of the pattern of expression. We have a GELS component to this project, looking at how gene therapy is perceived in society and how adoption might be improved for uptake of gene therapy. And Josh is working on our business development activities. And there's an awful lot of interactions on this project between communities of companies and researchers with an interest in particular patterns of expression. So we break our work down into two general areas. There's maxi promoters and mini promoters. And so, to put it simply, for the more molecular-minded, maxi promoters are going to be bats, and mini promoters are going to be plasmids or viruses. And so with maxi promoters, we take a large piece of DNA, we modify it slightly to introduce some of our key sequences, and then we take that piece of DNA and we're going to put it into our genome for further study. When we work with that, we're really trying to capture the endogenous expression of a gene. So we think that we're going to have all the sequences necessary to recapitulate most of the expression that's seen for the gene that is contained within the back. For mini promoters, we're going to go into a design process. We're going to try to select candidate regulatory sequences, and then we're going to put them together in small pieces. Moving forward for CanEuCre, that's basically less than 3,000 base pairs of DNA that we have promoter sequence. We recognize in doing that we're only going to get a subset of the regulatory sequences from the original gene, and so therefore we don't expect to reproduce the endogenous pattern expression. We hope to see that we'll either have a subset of the expression or at least perhaps a temporal portion of the expression. So let's focus on maxi promoters first, and then we'll move to the mini promoters. So these are some of the maxi promoters we did under the Pleiades promoter project. And what you see here is on the left are images from the Allen-Brain Atlas, where they're looking at the natural expression pattern of the gene in the mature mouse brain. And what you see on the right are the images that we get from our promoters driving the LAC-Z expression. And what you will notice here is that we're doing a reasonably good job of recapitulating the endogenous expression pattern. There's some deviations. For instance, if you look at the MAO-A gene, you'll see here that most of the expression observed in the Allen-Brain Atlas is more restricted, where we start picking up a little bit more of additional sequence. So sometimes we get a little broadening of the expression in the arm, but it's pretty close to what's observed for the endogenous gene.